It's a pleasure to present the next speaker. So we have here Professor Ron Libeskind-Hadas, who received his undergraduate degree from Harvard University in applied mathematics. And then he got a PhD in computer science from the University of Illinois. He's currently the founding chair of the Kravis Department of Integrated Sciences at Claremont McKenna College. And his research and teaching are mainly centered in the area of computational biology. So it's a great pleasure to have you here, and the floor is yours. I don't know if you can share the screen. Can you try to talk? Because for now I don't hear anything.
Let's try this.
Okay, now yes.
Perfect, okay.
Perfect, thank you so much.
Thank you, Blerina, for the very nice introduction. It's nice to see so many friends here, I'm thrilled to be with you. Thank you for having me. Let me check. Is audio okay? Perfect, okay. So as Blerina said, my name is Ron Libeskind-Hadas. I just go by Ron because the last name is long and complicated. Even my students can't use that, so just Ron is good. I am at the Kravis Department of Integrated Sciences at Claremont McKenna College, which is one of 5 undergraduate colleges in a consortium of colleges in Claremont, California, just outside of Los Angeles. So I want to begin by traveling backwards in time let's go to Charles Darwin, who wrote in The Origin of Species, "I can understand how a flower and a bee might slowly become, either simultaneously or one after the other, modified and adapted in the most perfect manner to each other by the continued preservation of individuals presenting mutual and slightly favorable deviations of structure." In other words, Charles Darwin is imagining how species may evolve in tandem, in what we call coevolution. And indeed, this is a widely understood phenomenon now. Charles Darwin was prescient, and this happens both in the mutualistic scenario that he was describing, where, for example, a flower and a bee both benefit from this relationship. And it also happens in other contexts, for example, in parasitic relationships, where there's a host and a parasite that are evolving in tandem, in something like an arms race, where the host is evolving defenses against the parasite, and the parasite is evolving mechanisms to maximize its utility of the host. So in any case, in both of these kinds of scenarios and in other coevolutionary scenarios, we're imagining a pair of species exerting selective pressure on each other and evolving in response to one another. And indeed, while Charles Darwin posited this and imagined this, which is really quite remarkable, it turns out that to study this carefully and systematically, we need to use phylogenetic trees, which of course Darwin himself was also prescient in his thinking about these. So let's take a look at a specific and fairly well-known scenario of mutualism. This is not exactly flowers and bees, but it's close. It's figs, which we see on the left, and their pollinators, wasps, fig wasps in particular. And lest you be alarmed by the size of that wasp, it's nowhere nearly that large. It's maybe a millimeter or 2 long. They're very small. But interestingly, these figs, the wild figs, and their pollinators are found in every continent on the planet, with the exception, surprisingly, of Antarctica. And what we see here is that there is a very high level of host symbiont specificity. And that's indicated here in these 2 phylogenetic trees. This is a partial phylogeny of wild figs and a partial phylogeny of their fig wasp pollinators. You can see the extant taxa at the tips. And this dotted line indicates that this particular species of fig wasp is the pollinator for this particular species of fig. And it's very highly specific, which is quite indicative of a coevolutionary pattern, but certainly far from proof. And so what we would like to do is understand whether these 2 phylogenies are actually likely to be co-evolving, and if so, what does that co-evolutionary history look like? And so over here on the right, you can see from this well-known paper by Jackson et al., a embedding, or if you will, a reconciliation of these 2 trees. And so what you see here is the phylogenetic tree for the fig species, ficus, and embedded in it, this phylogenetic tree for its pollinator. And you can see that in many cases where the fig lineages bifurcate, where they speciate, you see a concurrent co-speciation of the wasp lineage, which is indeed what would be consistent with coevolution. In fact, in perfect coevolution, you'd imagine these trees are exactly identical, and you can essentially map the fig tree perfectly onto the wasp tree perfectly onto the fig tree, such that every speciation event in one corresponds to a speciation event in the other. But nature is rarely quite so kind, and other events can happen, and here you can see some of them. So you see not only concurrent speciation, you see non-concurrent speciation, what we sometimes call duplication in this context, which is the wasp phylogeny splitting, but not at a split point in the fig phylogeny, but rather along a branch. You can also see these transfer events. These are host transfer events. And you can also sometimes see extinctions posited in these kinds of reconciliations. Here's another kind of poster child for coevolution. This one is a host-parasite relationship rather than host-symbiont, where on this side you see pocket gophers, and on this side you see the lice that infest them. And again, there's a very high level of host-parasite specificity, and we'd like to understand how those two species co-evolve by mapping one onto the other. Generally, it's the symbiont or the parasite that's mapped onto the host under the assumption that the symbiont is more responsive to the host than vice versa. So here is kind of an abstraction of the problem. We're given two phylogenetic trees. We'll call this the host, and this one is either the parasite or symbiont tree. And we have the mapping of the tips, the extant taxa, which need not be a bijection. It need not be one-to-one and onto, but it's just a mapping. I mean, many to one is fine. And we're positing in this model 4 different kinds of evolutionary events. And at the end of the talk, I'll mention that there are other events that one could consider as well, but we're just considering these 4. Co-speciation is simultaneous speciation of one on the other. So here you can see this tree, the parasite tree, embedded on the host tree, where this node E here is simultaneously speciating with this node here in the host. But you also see that E has a descendant A, which needs to map here according to the relationship that we've observed. And that incurs a loss event here because on this lineage here, there's no branching in the parasite, but there is one on the host. And so we posit that there was an extinction event, a loss. And over here, you see that in order to explain this mapping, we've also posited a host transfer event where when D speciated, one of its lineages transferred and started tracking on this lineage here. So this is one possible reconciliation. And you can see here co-speciations. You don't see any duplications here, but you see a transfer and you see a loss. Here is another mapping of the same pair of trees, but a different mapping. Here you can see that this node E speciates independently of a speciation in the host tree. We call that a duplication. And it's called a duplication because it's kind of analogous to a duplication of a gene in the species. In fact, the two problems of gene species reconciliation and host symbiont reconciliation are analogous computationally. And so here you can see that non-simultaneous speciation. Here you can see a co-speciation, loss, and loss. So those are 2 different reconciliations, and we would like to find, in this context, the most parsimonious solution. And so one possible way of thinking about that is co-speciation we would consider a null event. That's consistent with coevolution. So I might imagine that you want to minimize the number of duplication, transfer, and losses. So in this solution here, you have 1 loss, 1 transfer. That's 2 events. Over here, you have 1 duplication, loss, loss, loss. And that's 3 events other than co-speciation. So this one would be considered more parsimonious in that framework, assuming that we're trying to minimize the total number of duplications, transfers, and losses. But in fact, the problem is a little bit more interesting than that, because generally speaking, we might have weights associated with those events. Those weights are generally estimated, often inversely proportional to the probabilities or estimated probabilities of those events occurring. And so the framework is this. Given a host tree, T1, a parasite symbiont tree, T2, a mapping of the leaves of T2 onto T1, and event costs, we'll say non-negative event costs for duplication, transfer, and loss. We assume the co-speciation has a 0 cost, this is a null event, Our objective is to find a minimum cost mapping of the parasite symbiont tree onto the host tree. And that is the maximum parsimony formulation of this problem. So I have some good news and I have some bad news for you about this problem. The good news is this. If the host tree is completely dated, then an optimal reconciliation, one that minimizes that total cost, can be found very efficiently in polynomial time. This is a well-known result. It's been around for more than a decade. And by completely dated, let me just say a little bit more what I mean by that. We actually don't need the exact dates. We don't need the exact dates on the host nodes. We don't need nothing about the parasite symbiont. What we actually need is a relative ordering of the internal nodes of the host tree. And this is a little more subtle than it seems, because of course, a parent happened earlier than a child. But if you look at two different parts of the tree that don't have an ancestral relationship, for example, this node here and this node here, the topology of the tree does not indicate the relative times of those events because they're not ancestral to one another. So when we say a complete timing, what we really mean is a total ordering of the internal nodes of the host tree. We don't need the exact dates. Even that turns out to be very difficult to obtain in practice. So the good news is if we have that information, we can find an optimal reconciliation very efficiently. The bad news is we rarely have that kind of information. All right, but now let me tell you some more good news. The good news is if you're not modeling transfer events, only duplications and losses, and of course co-speciations in all event, then again, we can solve this problem perfectly in polynomial time, and we don't need any dating events. We don't need anything about dating. And I'll just foreshadow that the reason that transfers are tricky is because they're transferring a species from one part of the tree to another part of the tree, and that transfer is considered to happen in a given moment, at least in geological timescales, in time. And so we need to make sure that the landing site of that transfer is coexistent with the takeoff launching point of that transfer. And if we don't actually know the ordering of the events in the host tree, we don't know if this is a legitimate landing. And so we may posit landings, we may posit transfers that transfer a species to a lineage that doesn't yet exist or is gone. And that's why the transfer issue, post-transfer, is tricky. So the good news is without transfer, we can solve the problem in linear time. More good news is if you add the transfers back in, you can solve the problem in polynomial time. However, it turns out that those solutions that you get may be time inconsistent, as indicated in this kind of cartoon here. And so let me explain. What happened in this fragment of a reconciliation, I'm not showing you the whole thing, is that in part of the mapping of the blue tree onto the black tree, there was a host transfer to this branch here. And that may have actually been, although it was drawn upwards, it may have actually been consistent in time because this branch here may have been long enough to make this an actual horizontal transfer. But we don't know because we don't have timing information. And then that lineage descends here and made its own transfer back here. And you can see that in this fragment of the reconciliation, you actually have that this lineage, which is a descendant of this, actually had its ancestor happening earlier in time, which of course is ridiculous. It doesn't make any sense. And it's because, again, if we don't have timing information and we're positing transfers, we actually don't know anything about where they're going in time, and the time may actually be inconsistent in the solution. The great news is that even if we solve this problem, in other words, we don't worry about the timing issue, and we find an optimal solution, of course, one that may involve time travel, we can actually, after the fact, we can detect if the solution involved time travel. That's very easy to detect. So let me say this again. If you have duplication, transfer, and loss, and you want to find an optimal reconciliation, you actually can do it. However, the solution may cheat in the sense that it may posit a collection of transfer events in particular that are time inconsistent. The good news, though, is that after the fact, you can detect if that actually happened. And so that is good news indeed. In fact, I would call it great news. The problem is this. The problem is that if there isn't time inconsistency, there's really nothing we can do about it. We don't really know how to fix that problem. And so now, unfortunately, we've come to this bad news. Finding an optimal solution that is time consistent is in fact computationally intractable. It's NP hard, and our group and others have shown this. So there are a few alternatives that we can consider. One is to use fast heuristic methods that find good, but not necessarily optimal, time-consistent solutions. And there are a number of algorithmic approaches, heuristic approaches, for doing this. Some of them have been embedded into software tools. One of them is the Jane tool that came out of our group, but there's many other excellent tools that do the same thing. So these solutions are heuristics. They are guaranteed to be time consistent. Don't need to know the actual timing. They're just guaranteed to be consistent, to not cheat in time. But they may not be absolutely optimal. And the alternative is to find solutions that are truly optimal in polynomial time, but they might be time inconsistent. And there are many tools for doing this too. One of them is a tool from our group called Empress, which I'll mention again in a bit. So let me come back to the fig and wasp phylogenies. So here we have the result of a very significant multinational study that took place about a decade ago of figs and wasps. And here you can see there's something like 200 and some taxa in the fig phylogeny and about the same number in the fig wasp pollinator phylogeny. These are found in all continents other than Antarctica. After a great deal of field work, identifying these species and the association of extant taxa, we used our Jane tool to find a reconciliation, and there it is. It's huge. You can't really parse this, but let me just point out there's two trees going on here, a black one and a blue one. The black one is the fig, and the blue one is its pollinator. And from this study, a number of things were determined. One is you can see just faintly these horizontal, these host switching events here. And what was surprising is the number of these horizontal events, and they happened really across the 60 million years of evolution of this system. So that was one of the discoveries. It was a fairly robust result. Moreover, from that study, it was some of the patterns of dissemination of figs and wasps were determined. You see here the sort of the root here was not actually in the ocean. It's just that it's not known where it occurred. But this study of coevolutionary patterns allowed the PIs to understand quite a bit of new information about the migration patterns. And then coming back to the Galapagos, these tools have also been used to look at some of the host-parasite relationships of taxa found there. And these results, according to the paper, have implications for the conservation and taxonomy of the Galapagos mockingbirds. So it's nice to see computational tools being used in these kinds of capacities. In my last few minutes, because I want to leave ample time for questions, let me just talk about some important challenges that arise. So I mentioned that in this maximum parsimony framework, we have costs associated with each of the event types that we're modeling. In this case, duplication, transfer, and loss. And in the first example that I showed you, I was associating a cost of 1 for each of these events and just simply counting the number of events in the solution and trying to minimize that. But in truth, it's not really clear how these event costs should be estimated. And if you look in the literature, oftentimes what happens is papers are published where the event costs are simply the default values in the software system that was used to do the reconciliation, which is hardly a systematic way of doing business. So that's challenge number 1, and I'll talk about that in a moment. Challenge number 2 is, as we know, in many maximum parsimony frameworks for a number of different problems that arise in biology, there can be a very large number of equally good solutions. And those solutions may actually look quite different from one another. So an interesting and important question is, how do you make sense of the potentially very large number of equally optimal solutions so that you don't mischaracterize your solutions by simply choosing one needle from that giant haystack? So on the first question, how do we identify appropriate costs for the parameters, duplication, transfer, and loss? Let me tell you about a result that is due to a collaboration with several of my colleagues from about a decade ago. It's called EXSCAPE, and this kind of algorithmic framework has been embedded into our Empress tool for reconciliation. Here's the idea. Duplication, transfer, and loss are unitless parameters. They're just relative. The important thing is their relative scale, not their actual numerical value. So we can hold one of them, any one of them, at unit and just vary the other two relative to that. And so let's imagine that we in this case, we have we were going to have the cost of loss set to 1. And so now you have really two free variables, which is the cost of duplication. This is duplication was set to 1. So this is the cost of loss relative to duplication and transfer relative to duplication. And of course, there's an uncountably infinite number of values that those parameters could take. They could take any real number of value. But it turns out, and this is rather surprising, that in fact, those values fall into equivalence classes. Colors indicate those equivalence classes in this space here. And any two parameters in this space. So again, remember, this duplication was set to 1. And so this is the cost of loss and the cost of transfer. Any loss and transfer cost in this equivalence class will give rise to exactly the same optimal solution. There's no difference. There's no sensitivity to the solution in this region. And similarly for this blue region, any set cost of loss and transfer will give you rise to exactly the same set of solutions. Not same single solution, but the same set of solutions. And why that's nice is because previous to this, what would happen is frequently people would recognize that the solutions were sensitive to those costs, and so they would just randomly sample, and not very systematically because they didn't know what the effect is of sampling those costs. So they just randomly sample maybe 10 different combinations. But this framework allows you to say, well, if I sample one cost from here, one cost from each of these equivalence classes, I will have in fact sampled completely and perfectly all of the possible event costs. So it goes from countably infinite to finite, which is uncountably infinite to finite, which is extremely important. And it turns out that these equivalence classes can be computed in polynomial time, which is also, I think, a nice result. So this tool is now embedded into Empress, which is a phylogenetic reconciliation tool, and that allows users to systematically choose event costs. And oftentimes they actually have a sense of what region here is even reasonable to consider. And so that can even further limit the set of event costs that they need to consider. The second problem is that then even for any one event cost, there can be a very large number of solutions. So you can see here, like in this yellow region, it says that in the yellow region, any pair of costs here will give rise to the same solutions, but there are 16 of them. And this is a very, very small toy example. In fact, in many cases, it'll be many, many billions of solutions, as you can see here in a study that my colleague Mukul Bansal did in a species tree with 100 species and 4,700 gene trees. So this is now the gene trees are playing the role of the host or symbiont in the context of what we've been describing, that about 50% of the time there were more than 100 optimal solutions. In 15% of the cases, there were more than 10 to the 5th, and some had more than 10 to the 40th. And so that's very difficult to interpret when there are that many optimal solutions. So what do we do? One approach is to generate solutions kind of at random and just sample them and then present that sample. But it turns out we can do better. And so one of our results is this. We can actually compute the distances between all pairs of solutions in polynomial time, in spite of the fact that there can be an exponential number of solutions. And so what do we mean by that? Take a look at two different solutions. We have a distance metric that explains how different they are. And I'll leave the details of that for questions if you care to probe there. But imagine that you have some way of measuring the difference between two solutions. And now imagine you have a very large number of solutions and you would like to compute in this case, histogram of the pairwise distances. So this is this shows you that there are something like 1.5 times 10 to the 4th pairs that differ in 10 places. And it shows you that there are something like 1.5 times 10 to the 4th number of solutions that differ in 25 places. This distribution gives you a sense of the space of that optimal solution space. And this surprisingly can be done in polynomial time. And to do that, of course, you can't enumerate all the solutions explicitly because there's too many of them. That would take an exponential amount of time in general. But it turns out that by leaving the solution space in a compact graph theoretic representation, you can then operate inside that graph representation and get these solutions exactly, get this distribution exactly without any sampling. And so we've done this It's built into some of our software tools like Empress. It allows you to look at the distribution of the solution space. And we can see in some cases that there's these bimodal distributions, which suggest that perhaps there's clusters of solutions that are similar and different from one another. So this gives you some insight into that space and a little more confidence in reporting one solution. If the distribution suggests that actually there aren't that many differences between the solutions, then perhaps choosing one is reasonable. And if the distribution shows that there's many solutions that are quite different from one another, one has to be more cautious about what's reported. I'm going to close by just mentioning that we have explored other models. So I mentioned in this talk the DTL model, duplication transfer loss model, but we've looked at other models as well. For example, models such as DeepLore, and I think Joe Worth is here, and I saw Joe earlier. Joe actually was a contributor to this project as well, and helped us with some of the thinking on this. Looking at models in which we look at other kinds of events, including evolution happening outside of the species tree. I mean, you see here mentioning genetic rearrangement, because in this reconciliation framework, you can think of these not just as pairs of species, but you can also think about a tree being the host tree playing the role of a species tree, and the parasite symbiont, think of that instead as a gene tree evolving inside that species tree. And so these reconciliation problems apply not just for pairs of species, but also for species and gene trees. And then we've looked at other models where we add in coalescent processes and explore the computational complexity of reconciliation in those frameworks. And this is ongoing research. So future work in reconciliation, we're interested in extending this work from trees to networks, because of course we know that trees are not the only way of thinking about evolutionary processes. We're interested in exploring other kind of evolutionary events other than duplication, transfer, loss, and some of the others that we've described. And of course, ultimately developing increasingly more powerful software tools for biologists to use, because all of these algorithmic problems are extremely fun and exciting theoretically, but even more gratifying when they find their way into tools that can be used by practitioners. And with that, I am going to stop, and I'm happy to take questions, of course.